landed on an alpha-1 adrenergic receptor. All right? So we're talking about in the liver right now because we're looking at lowering the rate of glycolysis. So it happens to be that this is a GTP-coupled reaction, which means that this GTP sits there to provide energy. Like when this lands, you go to GDP plus PI, and you also get an energy release when that happens, and that energy provides the catalyst for all this to run. Right? You need energy to run something. So here you have this phospholipase C, which is sitting here attached to the alpha-1 adrenergic receptor. What happens is once this docking happens, right, once this happens here and you've got this energy, this splits into two different molecules, okay? One becomes a diacylglycerol release. So you've got a diacylglycerol, or a DAG, and I'll put that in parentheses, and some places use DG, this book uses DAG, so I'm trying to be consistent with the book. This becomes a second messenger. It travels to wherever protein kinase C is, all right, and activates it. The net result of multiple enzymes that we're not going to work into right now is that protein kinase C acts on all these enzymes. It increases glycogenolysis, so that there's more fuel available for export, and it lowers glycogenesis so that you're not storing this energy because if you've activated epinephrine, you need the energy somewhere else.
So,
now, go ahead.
The protein kinase C will work on enzymes?
Yes, so the protein kinase C works on other enzymes, and there are a lot of other enzymes, so we're just keep it short. Because for us, it matters more that we understand that we get to protein kinase C, and then you can go figure out where protein kinase C is activating. Because it's going to do a bunch of different stuff, and then we can talk about 8 reactions, so I'm just trying to keep it to the point. Okay, so over here, the other thing you end up having is, so it gets split into this phosphatidylinositol bisphosphate, right, 4,5 bisphosphate, okay, which gets converted to inositol 1,4,5 triphosphate, or IP3 is fine. IP3 acts as a second messenger because it travels to the endoplasmic reticulum and stimulates calcium release. Now calcium is another second messenger because it's moving from the endoplasmic reticulum to somewhere else, right? So you're just passing this signal on over and over again. So that's why we start talking about signal transduction cascades. It's not just one messenger. Now we've got 2 middle messengers, right? And you can have 3 or 4 middle messengers.
So a domino.
Yes, exactly. So here we've got another second messenger, which is now traveling to activate calcium calmodulin-dependent protein kinase A, which is a type of protein kinase A, but it's only activated or deactivated by the presence or absence of calcium. And then this acts on pyruvate kinase to lower glycolysis in the liver.
Okay, so the second messenger is the calcium.
There are 2. So you've got IP3, which moves to the endoplasmic reticulum, and you also got calcium, which moves to the calcium calmodulin.
So that will act as the second messenger too.
Yes. You can have multiple, and second messenger is kind of a misnomer because it's actually the third messenger, right? but
it works as a second.
Yeah, but we call it a second messenger because it didn't receive the signal from insulin,
Yeah.
or glucagon, or epinephrine, or it's just passing it on within the cell.
And then the calcium goes to the colomodron.
Yes, it goes to this big molecule, which is a calcium calmodulin-dependent protein kinase A, and activates that.
Yeah, and then why is the word calcium next to that? Just to show that it's going into it?
Because this is actually the
That's
whole
the
name
name.
of the thing.
Oh.
It's literally a calcium calmodulin-dependent
Okay.
protein kinase A. So that's why. And then, yes, it's just a protein kinase A, but only one that has this in front of it will be acted on by that. And then that acts on pyruvate kinase, which is another enzyme. So you've got an enzyme acting on another enzyme to lower glycolysis in the liver. So why are we learning these 2 big, long signal transduction cascades? A couple different reasons. But the big one is because they're good representative examples of how this works. When we get to lipids, we're going to be talking more about how some of these things are produced, which will explain more by why we need certain fats in the body, right? But right now, this is how, instead of me just going, your liver just got told by the pancreas that it needs to release more blood glucose because your brain's hungry, this is how that works. This is what the actual mechanism of talking is. So we're learning two different sets of reactions because GTP coupled reactions are really common and G proteins are super common. So now, once we get these, which takes a while, but once you get them, when you're reading a paper about something else and it's a G protein coupled reaction, you're like, oh, I remember what that is. Even if I haven't seen the enzyme before, even if I haven't seen the mechanism before, I know that general mechanism, and I could go back and look up what's actually happening, right? I can put the pieces together. So that's why we're choosing, and the book also chooses to focus on these two. And then insulin, like I said, we'll look at that later because it involves another set of signal transduction, but this is probably enough for one day. So let me take a picture of that.